So we're back. Screencast number 6, and this is going to finish up protein synthesis. This is the process of translation. Okay, translation is the actual synthesis of polypeptides on ribosomes. It depends on the complementary base pairing between the codon on the mRNA and now the transfer RNA, which is going to actually be called codon. If it's complementary, you have an anticodon or anticodon. All right, so let's go through the process. RNA translated into protein. All right, you remember our key players up here. You know, you have all three of them playing in at once: the ribosome, the transfer RNA molecule, see what it looks like, and the messenger RNA molecule made from the DNA. All three plays. Messenger RNA is the codon, the transfer RNA is the anticodon, and all right, so let's make sure we get that word in here. Anticodon, and that is your transfer RNA. Get great. It's important to know because those are the terms that are used. And so, how does this happen specifically? If you take a look at this picture here, we'll look at this picture here. The ribosomes bind to the messenger RNA which is in the cytoplasm, which came from the nucleus, and it moves along until it reaches its start codon, which you can see right there, the AUG, which I underlined from another year. AUG. Okay. Now, again, remember the AUG codes for an amino acid, which is this color on this diagram. It's the green in this diagram here. All right. So each anticodon or tRNA molecule contains the complementary amino acid for that codon, which came from the DNA code. All right. So as it continues to move along, okay, each tRNA molecule carries, as I said, the specific. Ribosomes make sure the chemical reaction catalyzing go on and forms the peptide bond between the two that we'll show you here. And then they continue to move down the line. Another tRNA comes in and the thing continues. All right. So there you are. There you see it in short step. Binds to the messenger RNA. tRNA brings the ribosome amino acid to the ribosome. The anticodon on the tRNA binds to the 3-letter mRNA momentarily. Ribosome creates a peptide bond that you can see in blue here between the two amino acids. So this is the catalyzing, the making of the polypeptide. So basically, translation taking place within. It's an easy process to understand. What's the overview? What's the big picture? Polypeptide or protein is made by the ribosome through the process of translation. All right. There are more pictures then coming in that we'll have here. So there you see the tRNA. Remember, the tRNA and the anticodon are one and the same. All right. The tRNA and anticodon. Transfer RNA brings the correct amino acid and again uses the anticodon. Notice it has the anticodon on it. So sometimes the whole molecule is called an anticodon, which basically isn't accurately a scientific. So the anticodon is on it. So recognizing using the base pair rule. Bam. So these tRNA molecules, there's not one for each amino acid. They can, whatever amino acid tends to bond onto a temporary bond onto the tRNA, that's the anticodon that's going to be found for it. All right. So an anticodon found on the tRNA, sometimes the tRNA is called the anticodon. All right. It's by itself. All right. Don't get caught up in the linguistics. tRNA, anticodon, mRNA, codon. All right. Take a look at a few pictures before moving on. Oh, no. We have an example first. So what I want to do, I want to let you get a chance. Or here's an example. I want you to do this. I want you to put this into your Cornell. All right. So right now, do it at scrap. So it says create an mRNA strand that would be coded for this strand here. So I'm going to stop this. Okay, so 5 seconds or so later, you have paused the screencast and you have done this, and this is what you have come up with. Now, again, you're going to need to be able to do this both in the formatives and, okay, so better to do. So there is the mRNA. So there is your DNA. How is it DNA? It's located by the T. T is always in DNA. U for either M or tRNA. But right now, your messenger RNA right here. All right, so let's ask a couple of questions from that messenger RNA. How many codons are represented by this code? So count the codons. All right. I'm going to use the pen here to count the codons. You've got 1, 2, 3, 4, 5, 6, 7, and 8. So the answer, there are 8 codons. Remember, 3 base pairs, a codon. How many amino acids would come from this? I'm going to stop while you think about that and write a number down. What makes sense that you already know? Use the chart. You should use the chart anyway. That answer, boys and girls, would be 7, because a UGA does not code for an amino acid. All right? And then the next skill that you would have here from this is to know what amino acids code for it. So normally by this, we put a line under it, and you put the amino acid. I'm going to write over these words. So you look up AUG is MET. We've been doing this in class. MET. What is UUU? UUU right there. You can use the bigger one. You can use the one in chapter 25. PAG. I'm going to try to get this done quickly. All right. GCG. Look for your G. GCG. GCG. GCG is below alanine. ALA. All right. I'm going to use regular letters because all right. I'll erase these out of here too and get letters in here because God help, I can't stand my typing. This one here. All right. QUU was PAG. Notice I'm reading from the table. Alanine. All right. Find UCU. Find UCU. You go below UCU, you'd find out that it's a serine. SER. All right. SER. Bring the alanine up. All right, find AAC. Find AAC. AAC. This is how I do it. I look it up. I'm not a big one for ASN. GCC. GCC. Find GCC. There it is over there. Wait. GCC again. Alanine. Hey, we had that before. So notice the code for more than one. GAU. GAU. Right there, and UGA, STOP, stop. That really is it. That's how you do it. All right. Let me use the code to find out the amino acid. All right. These amino acids then string together, these 6 or 7 there, to form a polypeptide. Unfortunately, that's 6 and 7 times about 5,000 more to form a polypeptide. All right. So moving on, you got pictures of the process. This goes translation a little bit more. Notice as you go further, we'll stop there for about a couple of seconds. You can see it, the growing polypeptide chain. These transfer RNA molecules can be reused. They just have to be recoded with a new anticodon. All right, forget about this picture. I'm going to take it out. All right, here you are, different picture, different kinds, same thing. All right, we're going to ask you to conclude here by also placing this example, do and place in the Cornell, but I am not going to do. And then we're going to compare cow insulin. I want you to compare, okay? Compare cow and human. And I want the answer placed in, excuse me, I didn't finish the spelling there, placed in the Cornell. Better listen to these things. I'm looking for these examples. Compare after it's done, compare the similarities in sequence of the number of amino acids. Are they the same? Are they different? How many are different? How close are a cow and a human to being same DNA? All right, that concludes